Today, I want to take you to a typical farmhouse in my village, a small village in the northwest region of Cameroon. You're welcome to Curious Molecules. If you're new here, you are very much welcome. If you're returning, please review, like, comment, and subscribe. All right, if you have never heard of a farmhouse or storage house in the village, this picture on the screen is going to give you an idea of what it is. Although it's not very detailed, but you can see that a bunch of corn cobs have been tied together and they're hanging there. Growing up, I saw my grandparents work very large hectares of farm, and I used to wonder how or what they're going to do with all those crops. They're talking maize, beans, potatoes, groundnuts, and so on and so forth. And the farms used to be so large that I was wondering how they're going to store up or eat up all this or even sell it. But in the village setting, for example, most people have their own farms, and you would notice that most people also work similar large hectares of farms with several crops and so on. So you wouldn't talk about selling or even sharing because everybody has excess. But the period of planting is a period of plenty, the rainy season where there is a lot, and people eat and eat and eat. But the good thing is that they don't forget to store up for the time when there will be less food. And so they had storage houses or farmhouses, which could either be in the farm itself or even at home. Storage houses where they will store excess of these crops. And one amazing thing was that the storage houses, you look at them physically, you wouldn't believe that all the crops are going to fit into that particular house or that particular room because there were some little huts, but they managed to find a way to maximize that space to make it very efficient. So, in order to maximize the space that was available, our grandparents used a method of storage where they don't just throw the corn cobs on the floor or in the chimney, wherever. They try to tie them up together in a bunch. The arrangement of those corn cobs should give you an idea of something, a carbohydrate molecule that you're seeing probably in the screen. And this molecule is what we are going to be discussing today. This molecule is glycogen. We are not just talking about glycogen today, we are talking about how glycogen is formed and why does the body have to produce glycogen. So, glycogenesis is what we are looking at today. Various molecules, anytime we encounter a new word, we always try to break it down into separate parts that will help us understand better. So here we have glyco and we have genesis. Glyco in relation to glycogen or glucose, and genesis, which simply means from the beginning or from start. So the formation of glycogen from scratch. So from breaking it down, you can already have an idea. Glycogenesis is the synthesis. This is not the first time we're mentioning synthesis, and synthesis is the same as production or manufacturing, whatever you want to put it. Synthesis of glycogen from glucose. And the major reason for synthesizing glycogen from glucose is to store it. Because in the analogy of the farmhouse, when farmers have harvested all those crops, they cannot eat all of it at once. Everybody has excess. You need to store it for the future. The same for our bodies. When you have just eaten your meal and it has a high level of carbohydrate, for example, you have just eaten rice and stew, rice and beans, rice and whatever, or yams, plantains, and all of that. So you have eaten a high carbohydrate meal, and probably you are not doing much work. Maybe you are just resting or whatever. So the carbohydrates are digested, they reach the small intestines, and then they go into the bloodstream. Remember from the video on digestion or from the lesson on digestion, if you don't know about it, you should go to the videos and watch digestion. So from the lesson on digestion, at the end of the day, in the small intestines, what is entering the bloodstream from the small intestines are monosaccharides, glucose, fructose, and galactose. So most times, if you're eating high carbohydrate foods, you have a lot more of glucose because, for example, starch is made of glucose and other carbohydrates or polysaccharides. So at the end of the day, you have when you're eating that carbohydrate-rich meal, the blood has excess glucose, and a hormone is produced. What is that hormone? If you said insulin, you guessed correctly. So insulin is produced by the pancreas, which tells the cells to take up the glucose from the blood because the blood does not want glucose to be too high. There is a certain level or a certain range in which blood glucose always alternate, cannot go higher or too low. Insulin tells the cells of different tissues to take up the glucose. And when cells take up this glucose, depending on the tissues, they will use it up, or if they have no need for it, they will have to take it back to the blood, and the blood does not need it. So the blood will send it to where it can be stored. And there are two organs in the body that can store glucose, the liver and the muscles. So the liver and the muscles can store all excess glucose, and they'll do that by the process of glycogenesis. In these liver and muscle cells, the specific organelle where this takes place is the cytoplasm or the cytosol, and also a certain level in the smooth endoplasmic reticulum. So we said insulin is responsible for telling cells to take up glucose. So the cells that are active, they are going to use it, break it down through glycolysis. If you don't know what glycolysis is or the pathway you want to understand it better, there's also a video on this channel on glycolysis. So the cells will take it, break it down through glycolysis and use it if they need the energy. But if they don't, then the liver and the muscle are going to convert it to glycogen. And this is where the body stores excess glucose. So that when time comes and you're starving, you're hungry, you've not eaten for a while, the body will be able to go back to those storage houses, which is the liver and the muscle, and tell them to bring back the glucose they had stored up, and they will use it to energize the cells of the body. All right, now that you understand why glycogen is formed and why it is even necessary in the first place, let us now see the process. So first, there are preliminary steps of glycogenesis. So like everything that you do, there is always a preparation before you do the actual thing. So in glycogenesis, there is a preparation step or preliminary steps. These steps are what prep the glucose to form glycogen. Remember, glycogen is just a bunch of glucose molecules linked together or that have formed bonds together. So this is what happens in the preliminary steps. First, when glucose has entered the cell, for it to go on to be metabolized, it has to be converted into glucose 6-phosphate. If you remember, there is another pathway that glucose is converted to glucose 6-phosphate. If you remember the pathway, please drop in the comment section. So glucose is converted to glucose 6-phosphate to trap it within the cell so that it does not diffuse back out of the cell. And glucose 6-phosphate is then isomerized to glucose 1-phosphate. So this is now taking it to another pathway. I already mentioned that there's another pathway that involves glucose 6-phosphate. So if you have glucose 6-phosphate available and it can choose any direction, then the molecule, if you want it to follow glycogenesis specifically, you have to convert it to something else. And so it is isomerized. A phosphate moves from carbon 6 to carbon 1. So there are many metabolic reactions that you will see where isomerization occurs. And I mentioned before in one of the pathways probably that there's a reason why the body does not want to use a lot of energy, but there are some steps that you might see or think that are very unnecessary. But the body does that maybe to prepare certain sites for the binding of enzymes and so on and so forth. So the body has its reasons for converting certain molecules to maybe isomers of themselves. You see the difference between one molecule and the previous one is just a single carbon and so on. So it's for binding and also to prepare the molecule for the next steps. So in this case, glucose 6 is converted to glucose 1-phosphate because the direction is glycogenesis. All right, the next thing that happens is kind of activation of glucose. So to activate glucose, UTP comes in, eliminates the phosphate, becomes UDP, and then binds to glucose. So you have UDP glucose at the end of the day. So UDP is basically there just to activate the glucose. So in forming glycogen, we also need a foundation. Foundation of glycogen here will be composed of a series of glucose molecules that are going to set the stage for the formation of the full glycogen molecule. And so there is a very important protein that is involved, glycogenin. The tyrosine molecule in glycogenin, tyrosine has an OH group. The OH group is going to interact with UDP glucose. UDP, which is found on position 1 of the UDP glucose, is going to leave, and where UDP was bound before is going to bind to the OH group in the tyrosine. The first glucose molecule has now bound to glycogenin. The next glucose molecules are going to do the same. An activated glucose comes in, the UDP leaves, where the UDP was on position 1, and so it now binds to the fourth carbon of the glucose in the brain chain. So you have UDP leaving from the first carbon or from carbon 1 on the activated glucose molecule. Then carbon 1 interacts with the carbon 4 in the glucose molecule of the growing glycogen chain. So that is what happens. 8 to 10 or 12 or even 20 molecules of glucose will form in this short chain to create a glycogen primer, which is like the foundation for the formation of the whole glycogen molecule. Okay, now that we have set the foundation for formation of glycogen, we can continue with the process. So the rest is easy. It's just the addition of glucose molecules to make the larger chain or the larger molecule. So the addition of glucose molecules is done by an enzyme called glycogen synthase. It's a very simple enzyme from the name glycogen synthase. You're synthesizing glycogen. So this is a very easy to remember enzyme. And glycogen synthase is able to add glucose molecules in a straight chain. So what happens is UDP glucose or the activated glucose comes in, UDP leaves from carbon 1, if you can see here. UDP leaves from carbon 1, makes carbon 1 free, and then carbon 1 can now form a bond with carbon 4 in the growing glycogen chain. So that's why you have alpha 1 4 bonds in a glycogen molecule. So that happens to elongate the chain or to make the chain long. But now, if you saw at the beginning of the class, the structure of the molecule of glycogen is not just a straight chain. There are branches. So how are these branches formed? When glycogen is being synthesized and a branch has to be formed, what happens is that the glycogen synthase stops working at that moment because glycogen synthase cannot form branches. Branches to be formed, another enzyme comes in. A simple enzyme, you can call it the branching enzyme, or the scientific name, which is amylo 1,4 to 16 transglycosylase. It basically explains the nature. The bonds are moving from 1,4 to 16. So the branching enzyme will remove a small part of it, around 6 to 8 glucose molecules long, and convert the bonding from 1,4 to 16. Remember, carbon 1 is bonded to carbon 4. So in this case, it will remove it from carbon 4 and take it to carbon 6, and you have a branch as is shown here. Bonds that are found in branches are alpha 16 glycosidic bonds. So this glucose molecule now has an open or a free carbon 4, where another glucose can bind to continue the straight chain, while this other part, the branch, also has this free carbon 4 at the last glucose molecule, where glucose or activated glucose can bind to continue the chain. This is what happens continuously till a full glycogen molecule is formed. You can be asking, why does glycogen need to be branched? We already talked about the analogy when we talked about the farmhouse. Branching is an efficient way of storage because we talked about maybe throwing the corn cobs on the floor like that, or maybe in a chain you need to store them. They'll occupy more space than if they were tied together in a certain arrangement. So it's the same for glycogen. To be able to store it efficiently, the branches are formed so that it occupies less space. Another very important thing about branching is that the branching allows glycogen to create many non-reducing ends. Non-reducing ends are those free carbon 4 ends or OH ends. The branching creates several of these non-reducing ends such that when the body needs energy, it can be broken down very fast because there are different ends for breakdown of glucose. The same for the synthesis too. It also makes synthesis very fast and efficient because there are many ends for glucose to be added. So branching is basically for efficiency of storage and efficiency of work for the body or for the cell. So the cell is quite wise, if you ask me. We are now going to another interesting part of this pathway, which is something that we discuss with every metabolic pathway that we have seen so far. We have seen that metabolism involves breakdown or synthesis of molecules, and all of these are controlled by the needs of the body. So if the body needs energy at a certain time, cells will not want to be synthesizing glycogen, for example. The body would make sure that the pathways, the metabolic pathways that are active, are the ones that are going to give it energy. But if the body has excess of, the blood, for example, has excess of nutrients, excess of glucose, then the body metabolic pathways that will be activated are the ones that can use up that glucose to free the blood from high glucose. And so we look at regulation to be able to understand how these particular pathways impact the body and to also better understand, especially with disorders that have a gene that affects one of these enzymes or affect one of these pathways and causes that person to have the symptoms that they have. So in this case, we are looking at regulation of glycogenesis. At the level of regulation, we have seen with other pathways, regulation targets key enzymes. Regulation targets rate limiting enzymes. Enzymes that when you modulate them, when you touch them, they are going to affect the whole pathway. The major enzyme here is glycogen synthase. We on the diagram you see it together with glycogen phosphorylase, but you will see that when we talk about breakdown of glycogen. And let's focus on glycogen synthase. First, there is allosteric regulation. Allosteric regulation, glucose 6-phosphate is an activator of glycogen synthase. Glucose 6-phosphate, we saw that is one of the preliminary steps of glycogen synthesis or glycogenesis. There's excess glucose 6 phosphate. The body is telling you that there is excess glucose, and so I'm converting it to glucose 6 phosphate. Glucose 6 phosphate is also a preliminary step in breakdown of glucose in glycolysis, and so when there's excess glucose, there's excess glucose 6 phosphate. And if there's excess glucose 6 phosphate, it means the body has excess glucose and you need to do something about it. If you're not using it in energy, then it has to be stored. And so storage means that convert more of the glucose to glycogen. So glucose 6 phosphate will activate the enzyme glycogen synthase. We also see that ATP and glucose would activate glycogen synthase, not directly. So they are rather activators of glycogen phosphorylase. We would see glycogen phosphorylase later. But if there's excess ATP, just think about it, right? Excess ATP, excess energy, excess glucose, it means that, okay, the body is not even using the energy, or it's too much as compared to the needs of the body. And so we have to convert the glucose that is available to something else, not energy, because I already have a lot. That's what the body is saying. All right, we are coming to another interesting aspect of regulation here, covalent modification. We have seen already that in the previous diagram there was active and inactive glycogen synthase and glycogen phosphorylase. We didn't talk about it, but we're talking about it here. The activation or inactivation of glycogen synthase is done by covalent modification. Covalent modification is basically when a molecule is modified by addition of a covalent molecule or by formation of covalent bonds. So glycogen synthase can form covalent bonds with phosphate. When a phosphate binds to glycogen synthase and phosphorylates it, it becomes inactive. And that inactive form is the GSB form, glycogen synthase B form, it's a phosphorylated form. While the active form is unphosphorylated or not phosphorylated, the phosphate is removed. That's covalent modification, covalent removal of a phosphate and covalent addition of a phosphate. So we already know that if it's active, it means that it's going to synthesize glycogen. If it is inactive, it means that there is no need for synthesis of glycogen. And so you can try to use hormones or glucose or even the glucose 6 phosphate that we talked about before. If glucose 6 phosphate is activating glycogen synthase, how will you explain it in terms of this covalent modification? For example, if glucose 6 phosphate is activating glycogen synthase, it means that it is causing glycogen synthase to leave from the phosphorylated form to the unphosphorylated form. These are just enzymes that catalyze the phosphorylation and the ones that remove phosphate. The phosphorylase A phosphatase removes the phosphate and converts the glycogen synthase from the inactive to the active form, while glycogen synthase kinase converts glycogen synthase from the active to the inactive form based on the needs. You also see some hormones that have been mentioned here, insulin, and here the red negative shows that it has a negative effect on this particular GSK3. So it will inhibit GSK3. GSK3, we have said, is glycogen synthase kinase 3, and it's supposed to add a phosphate. And if it adds a phosphate, what is it doing? It's inactivating glycogen synthase. So think about it. If insulin is high in the body, what is it trying to say? It's trying to say that there's excess glucose. So if there's excess glucose, I would need glycogen synthase to be active because I need it to synthesize glycogen and store it because I don't want excess of glucose in blood. So of course, insulin will be telling glycogen synthase to be active by causing it not to be phosphorylated, by inhibiting this enzyme that is supposed to phosphorylate it. So yes, and then insulin also can activate phosphorylase A phosphatase. If glycogen synthase is already in the inactive form, it can remove the phosphate because, again, we have explained if insulin is high, it's telling you that there's excess glucose and I need that glucose to either be used for energy or to be stored. So if the body has insulin can either inhibit the enzyme that is causing phosphorylation, causing glycogen synthase to be inactive, or it can remove the phosphate that is making it inactive and make it active. On the other hand, glucagon, for example, epinephrine, they are negative modulators or they are inhibitors of this phosphorylase A phosphatase. Let's take, for example, glucagon. When glucagon is released, glucagon is telling you that blood sugar is too low. My blood sugar is low, I need sugar in the blood. And so cells should stop using glucose for energy, or they should stop using glucose for storage, for example. So I don't want glycogen synthase to be active. So it will stop phosphorylase A phosphatase from acting, from removing the phosphate and making it active, because we don't want glycogen to be synthesized. We basically want to break down glycogen so that it can release glucose into the blood because blood sugar is low. So basically, try to reason the pathways like that, especially when relating them to physiological molecules or insulin, glucagon, hormones, and other molecules like that.